Hi Year 11s, welcome to the last PowerPoint and the last video for cells and microorganisms. This is probably going to be the last video for Term 1 as well, because we'll be working on a practical for the remainder of Term 1 before we start our next topic in Term 2. So in this lesson, we're going to be looking at microorganisms. Now, it's kind of a long PowerPoint and a long video, and it also is quite a long need to know, so I'd recommend if you want to write that down, maybe pause it at this point, and then you can write those down, but we're not going to stay on them too long. I've tried to make the slides a little bit smaller so that there's not as much to note as there possibly could be. But let's begin. So first off, I suppose, since talking about microorganisms, we probably have to define that term microbes. And what is the word microbes? So microbes are single-celled organisms. There are some that are multicellular, but they're mostly single-celled organisms. We're talking about things like bacteria, fungi, viruses, and protozoa. And when we're talking about multicellular microbes, we're talking mostly about fungi. Important point there, talking about viruses. Although viruses are classified as microbes, we're still not going to classify them as living things. It's really hard. They don't meet the Mr. Shen criteria that we talked about in week 1. Like I said, in that week there's a big debate with scientists whether viruses classify as living things or not, but for now we will consider them as not living things. We're going to look at each one of these in overview, so bacteria, fungi, viruses, and protozoa. Let's start with bacteria. So what exactly are bacteria? They're unicellular prokaryotes, so single-celled organisms. We can find them in air, soil, water, inside multicellular organisms like ourselves. We have millions inside our gut. What they do, basically, they're a very simple structure. Compared to eukaryotes, it's a lot simpler, like all prokaryote cells are simpler than eukaryotic cells. There are harmful bacteria, but there are also really useful bacteria as well. We classify bacteria based on their shape. So you can see here that there's 3 different shapes we need to know. So if it's a rod-shaped bacteria, it'll have the name Bacillus. If it's a sphere-shaped bacteria, it's going to have the name Coccus. And if it's a spiral-shaped bacteria, it's going to have the name Spirillus. Fungi. Fungi, these are eukaryotic cells, they're not prokaryotic cells. They have a nucleus. There are unicellular fungi, and there's also multicellular fungi. Example of unicellular is going to be yeast, whereas mushrooms are going to be an example of multicellular. Fungi themselves, they're heterotrophic. They don't have chloroplasts. They can't make their own food. They have to feed on other organic matter, which is what we're talking about here. Again, usually multicellular. Some are not microorganisms. Mushrooms, for example, are not microorganisms. That being said, if I ask you to look at a mushroom, draw a mushroom, you'll probably just draw the top of it. The mushroom is actually a lot more than what we actually see. There's a whole network of roots underneath the mushroom called hyphae, and they will help to draw in water and nutrients and help feed the organism. But that's a majority of it. The hyphae is the majority of the actual fungal mass. Yeast, single-celled fungi, it's microscopic. Most types of fungi are going to be somewhere between yeast and a mushroom. These are really the minimum. So minimum size is the yeast, and maximum size is the mushroom. Most of the fungi are going to be somewhere between that. As I said before, fungi don't have chloroplast or chlorophyll. They do carry out mitotic and meiotic cell division, and they do have cell walls. So it's not quite the same as an animal cell or a plant cell. It's kind of got features of both, and this is the same thing fungal cells. We don't need to draw them, but you just need to know. They don't have chloroplasts, but they do have a cell wall. Fungi will reproduce asexually and sexually, depending on what type of organism it is. So something like yeast is going to reproduce asexually because it's just going to go through mitosis to divide. Something like a mushroom will go through sexual reproduction by releasing spores, and then those spores will fertilize other mushrooms and we'll get new ones, and that also allows them to disperse and survive in different areas. Viruses, on the other hand, so the biggest thing that we'd say that viruses aren't going to be living things is they're not capable of independent metabolism. They have no mechanisms of their own for transmission from one host to another. On the other hand, viruses can be viewed under a light microscope. Can't be viewed under a light microscope. You need something like an electron microscope, which we will have here in the next couple of weeks, to potentially see types of viruses. Viruses, all they really are is genetic material surrounded by a protein coat. Ribonucleic acid, RNA, is their genetic information. What that does is when the RNA comes into your body, it disrupts the host cell's metabolisms and will convince that metabolism to synthesize a new virus particle. Viruses will differ a lot from living organisms. They only have one type of nucleic acid, which is RNA, and they have a crystalline structure. But the biggest thing, I suppose, is that they can't be controlled by antibiotics. So antibiotics aren't actually going to be able to help fight off viral infections. We've got the drawing here of two different types of viruses, but you don't need to be able to draw those. You just need to be able to recognize that there's two different types. Moving on to protists. Most protists, well, protists can be multicellular or unicellular, but most are going to be unicellular. They can be free-living or parasitic. Different types of protists, which are protozoa, they'll move in different ways. So, for example, amoeba, which is an example of a protist, it moves by projecting false feet made by what's called the protoplasm, and the remaining protoplasm will fold into their false foot. So they'll like move one foot down, and then the rest will flow into it. It's kind of like a slinky movement almost. Paramecium, on the other hand, will move by beating cilia that covers the cell, and that will allow them to move through fluids. Some will use flagella, such as trypanosome. Some protozoa themselves will reproduce by binary fission, while others will use multiple fission. So binary fission is when you split into two, but multiple fission is going to divide into more than two. That's mainly the microbes we need to focus on, but what we now need to talk about is how those different microbes grow. And for almost all microbes, cell division is what's responsible for the growth of the population. So, for example, bacteria grow through binary fission. Every 20 minutes, roughly, if the conditions are favourable, the bacteria will reproduce or will go through binary fission. Which means that every 20 minutes, if the conditions are right, that population is doubling, which means it's going to grow exponentially like we see in this curve here. Different bacteria are going to have different conditions for their growth. So we're looking at things like temperature, pH, moisture, nutrients, waste and removal. We're going to go through some terminology with those. So for example, temperature. There's different words we need for temperature. Minimum temperature is the lowest temperature at which a species will be able to grow. Because if they go below that, enzymes don't work efficiently. It's too slow and the enzymes don't work. Optimum temperature is the temperature at which growth rate will be the highest. So that's the ideal temperature for that organism to survive. Whereas maximum temperature is they can still survive at this temperature, but if they go any higher, the enzymes will denature and they won't grow at all. Same with pH. The pH scale, we can't see the whole scale here. If you do chemistry, you've seen it before anyway. But it ranges from 0 to 14. 0 to 6 is considered an acidic environment, 7 is neutral, and 8 to 14 is considered a basic or alkaline environment. There are variations in pH, so if you have a high pH environment, it can cause phospholipid molecules to decompose, breaking down the cell membrane. If the abilities of the enzymes and transport proteins are inhibited, they can be inhibited at low and high pHs, depending on what the optimal pH is for an organism. And acids and bases, they're changing the chemical structure of the nutrient molecules in the cell, which means that they might not always be able to get the nutrition they need because the pH level that they're living in has broken down the nutrients. Moisture. Water is really important for the structure and function of all cells. We use water for the cytoplasm. We use water as a method of temperature control because the temperature of water doesn't change much. It keeps constant. It takes a lot of energy to heat up by 1 degree of water. So it's optimal for growth if we use water. Water is great as a solvent. Most chemicals will be able to dissolve in it for chemical processes. Nutrients and wastes dissolve in water, then we can transport them through the cell membrane. So water is really important. In fact, for microorganisms like bacteria, etc., most of the nutrients they need will be coming from water that's dissolved or using osmosis to get into the cell. Nutrients. So what nutrients are we talking about that these bacteria or microbes need? So we've got carbon, nitrogen, sulfur, and phosphorus, minerals, vitamins, and oxygen. Carbon's used to make organic compounds. Most of the time we'll get that from atmosphere or organic matter. Nitrogen, sulfur, and phosphorus, these are all needed to build organic compounds. Organisms will get that from soil, atmosphere, and other organisms. Minerals required for certain enzymes to function, again from other organisms or soil. Vitamins required for certain enzymes to function, again they'll get them from other organisms or they can synthesize their own. Oxygen required for aerobic respiration. Some microbes don't need to go through aerobic respiration, but some do. If they need oxygen, they're just going to get it from the atmosphere. But water, we've said water is really useful, but we actually need to remove water. So microbes will generate waste products like any other organism. We talked about that the other week. And they need to get rid of that. Because if it accumulates, it can inhibit the growth of the population or of the bacteria cell itself. can cause damage to the organism. Basically, what we need to do is use our transport proteins, or the bacteria use their transport proteins, enzymes and other membrane proteins to facilitate the removal of that waste from the cells. Microbes being decomposers. So we're getting close to the end now. There's only 2 more slides after this. Microbes, most are considered what's known as a decomposer, and this is an ecosystem term we'll look at a lot more in term 2. But microbes decomposers, they're also so decomposers are essentially heterotrophs. They obtain nutrients for respiration and fermentation from other matter, from other organisms. They can't produce their own. But decomposers specifically will get it from detritus, which is dead matter, so organisms that have already died. So for bacteria and fungi, for example, what they'll do is they'll secrete enzymes. Those enzymes will digest the detritus. Once the molecules from the detritus have been broken down, the bacteria or the fungi will then absorb those nutrients into their body once they've been broken down, and then they can feed off that. Decomposers are really important for the ecosystem. Like I said, we'll talk about them a lot more in term 2, but they're important for now because they're linking the consumers and producers in an ecosystem and ensuring that there's a cycling of nutrients. Microbes are also important to humans. So obviously there's a lot of negative media about how all with microbes is dangerous, but in reality it's less than 1% of all known microbes are actually harmful to humans. Our large intestines, populated with large numbers of different bacteria. We use those bacteria to break down things like polysaccharides that our human body can't digest. More importantly though, well, probably not more importantly, but more in the media, we use bacteria for technology such as creation of artificial insulin on a large scale, which allows a lot more diabetics to be treated. We can take gene from insulin, place it in a bacterial cell, get the bacterial cell to replicate, and then we have large copies of that gene that creates insulin. We can then harvest that insulin, give it to diabetics. Also going to be useful for using wine making, bread making, beer making, etc. Microbes are important for all that. Food spoilage, and this is the last slide for the term. It's also quite interesting because this is what your pracs is going to be based on in the next couple of weeks. So microbes and food, they'll start the process of decay, they'll break down that food. You can't eat it at that point because you're at risk of food poisoning. So you can stop food from going bad by heating, freezing, drying, chemical treatment, or radiation. Heating, you're going to kill most of the bacteria during cooking, so thinking about chicken, you can't eat raw chicken because it's got salmonella in it. If you heat it up, salmonella dies because you're going past the maximum temperature they can survive at. Freezing, it slows down microbes, stops them from decaying the food, stops them from multiplying. You can't reproduce. So freezing is a good way of extending the life of food. Drying, removing all water. If you remove the water, it's a key function for microbes to grow. They can't go through their entire process of reproduction, prevent spores from germinating. It stops bacteria in the track by drying them. So dehydration is a great way to prevent food spoilage. Chemical treatment, so adding preservatives, and we'll look at that in a video a little bit more in class. And lastly, radiation. Radiation is used to kill microbes with gamma rays. The problem with that, though, is it can leave toxins in the food, which itself is dangerous. That's it for the PowerPoint. We'll watch this video in class next week, or this week. See you in class.